Hello everyone. Today we are diving into a high yield topic for USMLE Step 1, that is the choice of statistical tests for hypothesis testing. This is a topic that is going to show up in every USMLE Step 1 exam, so I need you to pay attention here. Before we talk about the statistical tests, we need to talk a little about the types of data. The statistical data can be broadly classified into two key types, that is the continuous data, also known as the quantitative data, and categorical data, also known as qualitative data. The continuous data is the data that can be measured. So how I remember that is that basically if you can answer how much about a set of data, then this is a measurable data. For example, we're talking about systolic blood pressure is 120 or 160. We can answer how much blood pressure, that is 120. In case of categorical data, the data falls into distinct groups or categories, but we cannot actually answer how much. For example, the blood type of a person is either A or B or AB or O. If the blood type of a person is A, we cannot answer how much A. It's either A or B or any type else. It's not how much A or how much B. So based on this classification of data, for continuous data, we have different tests, and for categorical data, we have different tests, and we'll talk about it further. For t-test and t-test and ANOVA are used for continuous data. What is a t-test? T-test is a statistical test that is used to measure or check the difference between the means of two groups. So when you have two groups of continuous data and you have to compare the means of two groups, your answer is going to be t-test. For example, to compare the mean of to compare the mean weight of boys versus girls in the class. For example, say there are two groups and we have to compare the means, and the data is measurable. We can answer how much weight a person has. So the answer is going to be a t-test. A t-test is the test of choice. So the t-test can be independent or pair t-test. These are the two key types. In case of independent test, the two test groups being tested are independent of each other. For example, we have to compare the weight of person taking drug A versus drug B, or we have to compare the weights of girls versus boys in a class. These groups are independent of each other, data is continuous, and this is going to be an independent t-test. Pair t-test is usually used for a before and after design. For example, we have to compare the weight of a group of a persons before taking the drug and after taking the drug. So the population is the same, group is the same, but we have to separate measurements, and this is going to be a pair t-test. On same, what happens when we have continuous data, but there are more than two groups to be compared? That's where ANOVA kicks in. This is an analysis of variance test, and this is used when we have continuous data, but more than three groups to be compared. So t-test is not applicable there, and we have to use another test, and that is where ANOVA comes. So remember, quantitative data, two groups, and we have to compare the means of two groups, then the answer is going to be a t-test, and we have to compare the mean of three or more groups. Now let's talk about categorical data. For categorical data, we have two tests, chi-square or Fisher's exact test. A chi-square test is, these are essentially more or less same, but the main difference lies in the sample size. For chi-square is used for large sample sizes, and when the question specifically mentions small sample size, then the Fisher's exact test is going to be used. We'll talk about in a little bit more detail. A chi-square is used to see if there is a relationship between two categorical variables. We have two groups, two or more groups of categorical variables, and we have to see if there is any relationship between them, and that is where a chi-square is used. Now, when do we make a choice between a chi-square and a Fisher's exact test? A chi-square is used when the expected then chi-square can be used when the minimum frequency in any group is 5 or more. If the minimum frequency is less than 5, then a chi-square is not used, and the Fisher exact test is used instead. Now, what you key takeaway for USMLE, a trick for USMLE is that if the question gives you a 2 by 2 table and your first take is a chi-square test unless otherwise specified. And what are the otherwise specifications is the minimum frequency in a group is less than 5, or the question exclusively mentions that the sample size is very small, then a Fisher's exact test is going to be used. Otherwise, chi-square is nearly always the answer. A Fisher's exact test is similar to chi-square, and that is used to compare categorical outcomes but in small populations. See, if the question gives a 2 by 2 table and tells you the small sample or any expected frequency less than 5, then a Fisher's exact test is used. Otherwise, see, this is a high yield table. Continuous data, two groups, t-test is going to be used. Right? Continuous data, more than two groups, ANOVA is going to be used. Categorical data, two groups, chi-square is going to be used with large population size. And categorical data with small sample size or smaller frequency less than 5, then a Fisher's exact test is going to be used. This flow chart here is your key takeaway from this session. If you do this, you will never get a question wrong on the choice of statistical test. First of all, we have to see if the data is numerical or categorical. If the data is numerical or measurable or quantitative, and we have to compare the means, then Fisher's exact test and chi-square are automatically out of question. We're left with t-test and ANOVA. Now, if there are two groups, then we are left with t-test. And if there are more than three groups, then t-test is not applicable. If there are two groups, now we have to see if an independent t-test or a pair t-test is going to be used. If there are two groups at one point in time, two independent samples, as we talked earlier, for example, a group patient taking drug A versus a group of patients taking drug B, these are independent of each other, and an unpaired or independent t-test is going to be used. There is one group at 2 points in time, two dependent sample, for example, population of patients before taking drug A and after taking drug A. So same population at different points in time, this is going to be pair t-test is going to be applicable. In case of three groups, and ANOVA is going to be used. You have to you have continuous data, three groups, and you have to compare the means. ANOVA is the answer. Now, if you have categorical data, t-test and ANOVA are automatically out of question. Okay, so Now we have categorical data, and we have sample size or the minimum frequency less than 5. A Fisher's exact test is going to be used. Otherwise, for a large sample size or minimum frequency less than more than 5, and we have to compare two or more groups, का इसका response.